Gut microbiota, a fascinating community living inside us.
Welcome to the fascinating world of gut microbiota. Today, we'll dive deep into understanding these microscopic inhabitants of our gut, their importance, clinical implications, metabolic functions, and the revolutionary concept of gut microbiota transplant. Our gut is home to trillions of microorganisms, including bacteria, viruses, fungi, and protozoa, collectively known as the gut microbiota. The gut microbiota plays a pivotal role in digestion, helping break down complex carbohydrates and fibers that our own enzymes can't handle. But it is beyond digestion. Gut bacteria in the cecum and large intestine produce short-chain fatty acids mainly from non-digestible carbohydrates that pass the small intestine unaffected. These fatty acids improve gut health through maintenance of intestinal barrier integrity, mucus production, and protection against inflammation to reduce the risk of colorectal cancer. They're our first line of defense against pathogens, preventing harmful bacteria from taking over. Interestingly, they also train our immune system, teaching it to distinguish between friends and foes. Research has shown a profound connection between gut microbiota and various health conditions, from inflammatory bowel diseases to obesity and even mental health disorders like depression and anxiety.
The stomach hosts a little microbial community due to its acidic environment. The small intestine has a more diverse microbiota. The ileum has a higher microbial density and diversity, including members of the Bacteroidetes and Firmicutes phyla. The large intestine harbors the most dense and diverse microbial community in the human body, with estimates of over 1,000 species. The environment in the colon is anaerobic, which supports the growth of a wide variety of bacteria. The main functions of the colon microbiota include fermentation of undigested food residues, synthesis of vitamins, e.g., vitamin K and some B vitamins, and regulation of the immune system. Infants are born without gut microbiota, but rapidly after birth the infant's gut is conventionalized by bacteria coming from the mother and the surrounding environment. The composition of the microbiota stays unstable until the age of approximately 3-4 years, when it becomes mature. These microorganisms metabolize undigested carbohydrates, synthesize vitamin K and B. A significant portion of the body's serotonin is produced in the gut. Certain gut bacteria can produce tryptophan, a precursor to serotonin, affecting mood and gastrointestinal motility. Diets high in sugar and processed foods, and low in fiber can decrease the diversity of the gut microbiota. These foods promote the growth of certain harmful bacteria, and can lead to dysbiosis. Overuse or misuse of antibiotics can harm the gut microbiota by killing beneficial bacteria along with harmful ones. This can lead to an increase in antibiotic-resistant bacteria. Regular physical activity has been shown to increase the diversity of the gut microbiome. Sedentary lifestyles, chronic stress, alcohol consumption and less sleep can lead to a less diverse microbiota, potentially increasing the risk of inflammation and disease. Gut microbiota transplant, or fecal microbiota transplantation, FMT, involves transferring gut microbiota from a healthy donor to a recipient, aiming to restore a healthy microbial balance. The gut microbiome can influence estrogen levels through the production of an enzyme called beta-glucuronidase, which modulates the enterohepatic circulation of estrogens. This process can impact estrogen-related conditions like endometriosis, breast cancer, and osteoporosis. The hypothalamic pituitary adrenal HPA axis, which controls the stress response, can be influenced by gut bacteria. Certain bacteria can modulate the release of stress hormones like cortisol, potentially affecting anxiety and depression symptoms. Alterations in the gut microbiome have been associated with polycystic ovary syndrome, cause obesity, and type 2 diabetes, conditions that have strong hormonal components.